Start. All right, I think we'll get started with the keynote presentations. And the advantage of being the person introducing the keynoters is that I'll have a table to eat my lunch on. The organizing committee thought that we ought to pick for keynotes areas that we thought were rapidly changing right now and might deserve even more time. So we picked two topics. And the first one is the changes that are occurring in immunologic approaches to cancer. And of course, that's huge. It includes antibodies and vaccines and educated cells. And we're fortunate today that Carl June has agreed to be the speaker. He's currently the Director of Translational Research at the Abramson Cancer Center at the University of Pennsylvania. And he's studying ways to activate lymphocytes in order to modify or accelerate the immune response using the technologies of modern molecular genetics. Carl, welcome.
Well, thank you, John. It's a really great honor to be able to do this, and I think it represents a tipping point for immunotherapy that it's really now becoming integrated with other kinds of cancer therapy. And there has been a disappointing start and long runway, really, for immunotherapy, and a lot of skepticism that's been duly recognized. I think now we're at a tipping point, and I'll try to give an overview or a 50,000-foot view of some lessons from immunotherapy and how they address today's topic. So I have conflict in that the technology that I'll present, some of it is licensed by Novartis and is being developed now commercially, and that the principal investigators do not have conflicts. So there are a number of issues we've wrestled with this morning about how tumor evolution confounds and makes it very difficult to really treat cancer, and very much more now more like the HIV issue of hitting a moving target. And I've listed a bunch of these here. And immunotherapy is different in that it attacks the immune system, and it's not necessarily directly at the patient, so that many of these issues could be circumvented with successful immunotherapy. When you look at the field of immunotherapy, there are numbers of different modalities that are already used. Antibodies came in the 1990s, and we have their first therapeutic cell-based vaccine in 2010, Provenge, for prostate cancer. Cytokines have been around for a number of years. And cell-based therapies that are genetically modified are just now coming to the forefront. All these will interact with each other. In general, chemotherapy targets the tumor, and immunotherapy, at least with natural vaccines and so on, targets the immune system. Although the kind of genetically modified cells that I'll talk about are a synthetic form of biology that actually target directly the tumor. But many immunotherapy approaches target directly the patient's immune system and overcome immunosuppression. And radiotherapy, it's realized recently, can now target both, the so-called abscopal effect that Judd Walczak described last year in melanoma following radiation therapy. There are three basic laws of immunotherapy that apply that for cancer biologists, if you just remember these three, you'll be okay. One is that there's many different drug targets that the evolved receptors of B and T cell receptors can recognize. So virtually anything because of the recombination of the vertebrate immune system. The second law, though, is what's held immunotherapy back, which is that there have been numerous evolved mechanisms to overcome immune therapy against a tumor, which because tumors are similar to the basic normal tissues of our bodies. And so these checkpoints are those that prevent, in general, autoimmune disease. And I'll present two main approaches to overcome tolerance, one being so-called checkpoint therapy, and the other the use of genetically engineered cells. The final law that needs to be recognized is that the response to the same antigen is context dependent, so that if you have an antigen, and in the GI tract, say a gastrointestinal malignancy, the same antigen in the skin will have a different immune response. So whereas in the GI tract, it will be dominated with TGF-beta and tend to drive one towards an antibody response. In the skin, you'll get an inflammatory response. And so that the immunotherapy approaches for melanoma will be different for GI malignancies. So another realization now is that chemotherapy will interact in many cases in a positive way with immunotherapies to overcome tolerance. And on this slide, there's a number of approaches that are used to overcome the second law of immune tolerance. I'll describe the use of anti-CTLA-4 and PD-1 as checkpoint blockade. At the bottom, there are 3 different approaches with adoptive cellular therapy, which is basically a transfusion approach to use various forms of engineered T cells. And chemotherapy has been realized recently has and can have potent effects at overcoming immunosuppression, in particular at driving effects on the myeloid-derived suppressor cells and regulatory T cells. So they can shape, the chemotherapy can affect both the tumors and the tumor microenvironment. And how we interact all these together is a large challenge. There's schedule-dependent effects and dosing effects. The NCI about 5 years ago started a CITN, a Cancer Immunotherapy Treatment Network, led by Mac Cheever out of the Fred Hutchinson Cancer Center. There's about 28 centers, and they have held meetings to prioritize immunotherapy agents for vaccines and cytokine-based therapies. And they've been prioritized on a number of issues, such as whether it's a driver mutation target or a truncal target, various cytokines that have better effects than what we currently have available. And the issue this group faces now is the ability to do combinatorial trials. This is just probably our central issue because it's been very difficult to do this due to access to numbers and of experimental agents, as well as the difficulty of getting biologic trials started at multiple centers. So the first push towards personalized therapy happened in 2010 when the Provenge dendritic cell vaccine came out, and Nature Medicine shortly thereafter had an editorial on how personalized cell therapies might get, because many trials had attempted to make cell-based vaccines. And this is, in the case of Provenge, it's an autologous dendritic cell loaded with prostate-specific fusion antigen, and then given to patients as an intravenous infusion 3 times over 6 weeks. And there have been both opponents and proponents of this therapy. It gives a 4-month survival advantage over standard docetaxel, but it's not been shown to have tumor regression. It causes a survival advantage, so the mechanism's been hard to establish. And in addition, the manufacturing has been difficult to make those dendritic cells. Some regulatory conditions are shown here, considerations. For most of oncology, it's the left-hand side when you do a first-in-human trial, where one determines a Noel dose and tests in several animal models, and then does a 10-fold scaling factor to get the recommended starting dose. For biologics, in particular the cell-based therapies that I go through, are done by CBER rather than CDER, and they are done in the Office of Cell and Tissue and Gene Therapy. So there's a whole group now at the FDA just for these kinds of therapies. And there, in our first-in-human trials, we don't start at we drive a different dose. It's called the MABEL dose, the minimally anticipated biologic effect level, and begin just below that and test around that rather than trying to determine a maximum tolerated dose using the approach for small molecules. There are a number of considerations for replicating drugs. The T cells that we give to patients that have been genetically engineered have now persisted for over a decade in our first trials. So the projected half-life of an engineered T cell in patients with HIV, where we first start, is more than 17 years. So essentially, they're projected to have a stem cell-like persistence in patients. So that makes it difficult to do a phase 1 dose escalation study when you have a replicating cell. So we try to find a threshold dose and not an optimal, a maximum tolerated dose. So metabolism is very different, where we have both the cells are administered, then there can be a proliferative phase, and a maintenance and a memory phase, and so very different than the PK that one sees with a drug that may have standard pharmacology. So that means the trial designs are very different, and considerations of optimal biologic dose compared to maximum tolerated dose. So gene-modified cells are self-replicating, and that's both their promise and also leads to different safety considerations, and which we could talk about. So rather than vaccines, which just try to regenerate the immune system or rejuvenate it and overcome tolerance, another approach is a synthetic biology approach, which is to make cells that didn't ever exist in the first place in the immune system, and in this case, either using engineered high-affinity T-cell receptors that are not normally present in the human repertoire. And so these are HLA-A-restricted receptors that can be introduced to a T cell and then create a bispecific cell. The other approach is a so-called CAR approach, or chimeric antigen receptor approach, on the right. And that is the introduction of a fusion protein that combines an antibody binding domain, which then combines to the exterior of tumor targets or any target cell that has that target that the antibody fragment binds to, and then an intracellular signaling domain. The advantages of the CAR approach are that it's more off the shelf in that it's not restricted by the patient's HLA type. So the T cell receptor has to be matched to where the original patient's T cell receptor came from to the one that you're using as a transgene in the recipient patient. And another major approach is that it's turned out many human tumors have loss variants at the time of diagnosis where they've escaped the immune system. It shows that there's a natural immunosurveillance that's ongoing, and many tumors have lost antigen expression, usually through loss of HLA expression on the surface. So it's not uncommon in about 50% of ovarian cancers, for instance, to have MHC class 1 loss variant. And then a T cell can never kill you. So T cells can only kill if you're a natural T cell if you express HLA class 1. So we don't find, for instance, T cells ever cause an autoimmune hemolytic anemia by directly lysing red cells because they don't have HLA class 1. You can find antibodies doing that because they can break out and break up a red cell. So the antibody-based CAR approach then can kill these tumor cells that have lost class 1 molecules, and that's a major advantage over the natural immune system. So this is in a little cartoon of how CAR cells work. We introduce into a natural T cell receptor a transgene, the CAR that I described, in general using either a retrovirus or a lentivirus, so that's then integrated in genetically into the T cell and will be there. And then that's expressed constitutively on the surface of the cell, so that now the cell is bispecific. It has its endogenous T cell receptor, which may have been, for instance, a influenza-specific T cell, and it has now the CAR, or the antibody-based receptor in it, and it then can continue to exert its functions as, say, a flu-specific cell, or in this case, one that will kill any cell that expresses CD19. The major advantage of this probably is twofold. One is you don't have to wait for a bunch of serial boostings, as in natural vaccines, but the cells are infused all in one fell swoop. And secondly, they're non-cross resistant. So T cells have been shown to kill in very different ways than chemotherapy, and which is probably the reason that bone marrow transplants, when they work, have long-lasting effects. It's through the T cells that come in and give non-cross resistant and long-lasting effects on tumor cells. So we began our initial trial in 2010 treating patients, elderly patients with CLL. And all 3 patients who had late-stage incurable CLL, 2 of them with P53 mutations. And they had large bulk tumors of 3 to 7 pounds of tumor, and the tumors went away. And we could show that the T cells proliferated and that each T cell quantitatively eliminated 1,000 or more tumor cells, the so-called then serial killer cells. We now know 3 years later those remissions are durable, so that 2 of the 3 patients who had CRs remain without detectable leukemia, and they also have CAR cells that are persisted. And they have no normal B cells. They have B cell aplasia because the target CD19 is also on their leukemia cell. And so there's durable effects. And we've recently, over the last year and a half now, began begun to treat childhood pre-B-cell ALL, which is in general cured with high doses of chemotherapy and oftentimes radiotherapy. But secondary relapse CLL has a survival very similar to pancreatic cancer. So it's still the #1 cause of pediatric cancer mortality. And those are the patients we started treating about a year and a half ago now. And at this point, we treated over 50 patients at the University of Pennsylvania, and 17 have had of children with ALL, with many, many previous therapies, as you can see here, 3 to 8 prior therapies. 11 had actually relapsed after an allotransplant, and there's never been a therapy that works in that setting. And we treated 5 adults now so far, and with refractory ALL, and 4 of 5 have had a CR. And this is the response rate in our pediatric ALL trial, where there's been an 82% CR rate in these multiply treated patients. So the first patient was treated in April of 2012 and now is out past 18 months. So a high CR rate. We've had 2 patients who have had what you would predict, which is the loss variance. We've had 2 relapse with CD19 negative tumors. So that we're going to end up needing combinatorial approaches. But interestingly, in CLL, we've not had any patient relapse with a CD19 negative tumor. So anyway, we're seeing very high response rates. These are early, need to be followed up. The main side effect is cytokine release syndrome, and which is on target, and B-cell aplasia, which is managed by IVIG replacement. So at this point, between the 55 patients we've treated, there's been a 72% response rate. The issue is, how can we get beyond boutique cancer centers and bring this out into the public in a large center? And that's one of the challenges we have with these therapies. And we need policies that will promote the infrastructure to do cell-based therapies, and I'll discuss this. So we need to go from just pharmaceutical, biotech, pillars and medical devices to adding on a therapeutic pillar of delivering personalized cell-based therapies. There's a number of issues on these, such as that these are all N of 1, meaning at the FDA level, they're each one individually released. Some of these kinds of approaches will happen peripherally in a blood bank-like model, like the Red Cross, and manufacturing will be done locally, and others will require manufacturing at central manufacturing plants. Novartis purchased a plant last December and will open multi-center international trials in these leukemia cases that I was showing sometime in 2014 in order to get further experience in a phase 2 setting. One interesting lesson on how this can happen is called, it's not scaling up, but it's called outscaling. And in bone marrow transplantation, when I trained at the Fred Hutchinson in the early 1980s, they said we could never do bone marrow transplantation beyond boutique centers. And they just this year have celebrated the 1 millionth bone marrow transplantation. And these were done all one by one, but by outscaling, which was teaching other places how to do it. And that's how we do renal transplants and heart transplants at this point. They've gone from specialized centers out to more and more peripheralized centers. So there'll be both of those kinds of approaches to make this a reality. There are a number of challenges with these cell-based therapies shown here, such as there's been a large divide, a valley of death, because until just recently there was no biotech or pharma support. These kinds of immunotherapies all happened in the academic arenas. And they're difficult to conduct these trials in academic centers because the clinical trial costs have to be raised. Basically, there's usually not a drug company there paying for this, and the academic physician teams need to raise both the money to make manufacturing costs as well as the clinical trial costs. And so until just recently, there was no pharmaceutical involvement to push these things forward to phase 2, and they've all died after a phase 1 study and never been gone forward. So this just sums up. In 2010, we had Provenge, first FDA approved, and then 2011, the first checkpoint inhibitor, Ipilimumab. And on the horizon now, probably next year, we have Nivolumab, which is PD-1. And these are going to revolutionize solid cancer therapies. So there's a whole CD27B7 family. The therapy that I described with these CAR T cells uses CD28 as an agonist. CTLA-4 is a shutdown molecule, so it's in red, and so is PD-1. It's part of this checkpoint blockade route, and they're all evolutionarily related, these molecules, but they have diametrically opposite functions when expressed on T cells. CTLA-4 antibodies, or Ipilimumab, act to turn back on a response that was shut down, and the CTLA-4 ligands expressed in lymph nodes, so that when we give Ipilimumab, it's working in general peripherally, but also some in centrally. Ipilimumab then has an activity overall of 10 to 15% response rate in metastatic melanoma, but there's a tail on the curve, and then about 20% of people have at least 3 years of disease control. So we have an instance where the survival rate is greater than the response rate. There are a number of challenges with IPI that hasn't combined well with chemotherapy. If a vaccine alone is given, it actually decreases survival. So that was shown in the registration trial that Bristol Myers conducted for Ipilimumab. There have been separate toxicity issues in the so-called immune response, or IRAEs, that are very different than the kinds of toxicities that are experienced with cytotoxic agents. And another issue with IPI and these other biologics right now is that there are no good predictive biomarkers to know who's going to respond. We have to treat 5 people to have 1 person respond, but those responses are very durable at this point. As I mentioned, there's been toxicity with Ipilimumab that in some ways looks like autoimmune disease, with the most dose-limiting issue being colitis. But other severe endocrinologic issues such as hypophysitis. And it's very important to note that the management of, say, of a diarrhea in a patient on Ipilimumab is very different than in a patient who's getting a cytotoxic chemotherapy. So with cytotoxic chemotherapy, very often something such as loperamide would be given, or Lomotil, and in the case of the colitis with Ipilimumab and diarrhea, it's corticosteroids are given. So we need to train oncologists on how to manage immune-based adverse events rather than cytotoxics that they're known. Now, this spring in ASCO was a major watershed, I think, with the data that came out from several groups on PD-1. So PD-1, as I mentioned, is another checkpoint inhibitor, but it's expressed more, its ligand at the tumor microenvironment rather than in the central lymph nodes, as in Ipilimumab target, which is CTLA-4. So there are now several agents to interrupt the PD-1 blockade, or axis, one being the drug Nivolumab being developed by Bristol Myers, and the other blocks PD ligand 1, and that's Merck is testing that, and there's a number of these in various stages of development. The data at ASCO this year in metastatic lung cancer was quite striking, in that there's a dose response rate and a very long median disease control durations, over 83 weeks at the highest dose tested, and with very minimal toxicity in patients with metastatic non-small cell. And this is the actual data that was published in the New England Journal of Medicine this year, with a very significant tail and ongoing durable responses. And I think that it's possible to say that in a few years, immunotherapy will be frontline therapy for small cell lung cancer. I think that's an amazing watershed event when you look at this data and then how it will be combined with other agents. So we're at a really big tipping point in these agents. So we're going to be looking at giving combinations of therapy. On the left here is Ipilimumab, where there's 20% long-term survivors in metastatic melanoma. If you give vemurafenib, you get a very good acute response to people who have the V600E mutation, but then they pretty rapidly develop resistance. So the goal of the field now is this idea that we can use targeted agents to get the initial survival benefit, and then to add in immunotherapies that may get the tail effect, and then finally, hopefully have very long-term and high rates of disease control, and hopefully cures with combinations of immunotherapy and targeted agents. One of the first examples I've seen of this is from a colleague of mine, Cassian Yi, who, looking at this molecules here of different immunosuppressive molecules, CTLA-4 and PD-1. Cassian has treated a patient with metastatic melanoma with Ipilimumab, and he gave adoptively transferred T cells, which alone did not induce a complete response. But when he chased that with Ipilimumab, the patient had a complete response. So this is exciting. It's what's been modeled in preclinical animal experiments. So we will be seeing combinations of checkpoints with T cells and checkpoints with targeted therapies. It won't all be easy. This is an example that was published in the New England Journal this year where vemurafenib and Ipilimumab had unexpected hepatotoxicity, and the trial was closed. So that wasn't predicted by preclinical modeling. One of the major limitations of these multi-center studies is biospecimen analysis, which is more complicated than many targeted therapies where nucleic acid can work. So the best assays oftentimes are with live cells, and those haven't been implemented in multi-center studies, oftentimes sophisticated immunologic studies as well as a standard target assays. So I wrote down here a few issues that are policy issues and challenges. One being that multi-center trials with personalized cell and gene-based therapies are difficult. There's really no precedent for this in the cooperative groups because they haven't grown up doing this. So it's a new area, and the infrastructure needs to be established to do this. One of the particular issues is the manufacturing and liability issues with cell and gene-based therapies. If it's done at one institution, will another institution indemnify the other? And how are we going to do this? Right now, I can tell you if you're a state institution trying to negotiate this with a private institution, such as my university with a state university, it's really hard. They don't talk the same language, and there's different mechanisms of indemnification. We have changing paradigms for the toxicity management, as I mentioned, for instance, steroid versus low modal on diarrhea. So we have a huge issue on educating physicians on the principles of immunotherapy, and when the physicians that we have in this room went to medical school, I can say probably that what we got in our education in immunology was not much, and certainly nothing about cancer immunotherapy. And we have issues on trials design that I mentioned, very different for cytotoxics versus biologics. And I think we will have bucket trials for these immune-based therapies as well, and those issues are really in common as what we heard a lot about this morning. The regulatory environment is more difficult for cell-based and gene-modified therapies than it is for cytotoxic go through CDER. So if we have using an integrating viral vector, need to do a RAC OBA submission, so that's something that happens. There's 4 times a year there's a deadline, and we have to submit a protocol to get RAC approval as well as all the other FDA and IRB approval issues. And I think one large issue, policy issue facing our field is we have no incentive to develop immunotherapies for cancer prevention, which is where we would have the largest effects and impact. So for instance, we have a BRCA1 deficient clinic, for breast and ovarian cancer at the University of Pennsylvania. Those would be ideal patients to have on immunotherapy preventative trials, but they take too long with current patent expirations and so on for companies to have incentivization to do that. So I think that's a major policy issue to take on. So I'd like to end with that, and thank you for your attention.
The talks are quite different, so if there's questions about the immunotherapy, why don't we take them now, just a few questions. That was a wonderful summary of how this field has changed in 3 years.